specializing in sarcoma. He plays an important role in the design and implementation of MD Anderson's clinical next generation sequencing strategy. Today, he's going to talk with us about pathology and how best to navigate tissue decisions in the context of a sarcoma diagnosis. Dr. Lazar, the floor is all yours.
Okay, perfect. Can you hear me?
Yes. We can.
Okay, perfect. All right. So first of all, you can see that a really old picture of me. That's actually is the official picture of me from the MD Anderson site, but I don't really look like that anymore, as you can see. But that's okay. It's good to remember what was like in days gone past. Are we ready for me to share screen?
Yes.
All right. And I think this will be the right one. Okay. Are you guys seeing my whole screen now?
Yes.
Okay, perfect. All right. So so I'm going to talk to you about sarcoma pathology today. And my talk is sort of trying to give you some insight into what we do. So this is Sarcoma Pathology Unmasked, or why do I get a bill from a doctor I never met? You already saw the introduction from me. So before I get started, first of all, I want to say Happy New Year to everybody. I'm Jewish, and we just started celebrating the Jewish New Year, and good luck to everybody that's heading into Yom Kippur. So let's get started here. So I'm first going to talk a little bit about my side passion project that I do. I produce theater in both off Broadway in New York and also in Houston. This is the first thing that I actually produced in New York that I was actually recognized. It was nominated for a drama desk, but I ended up getting beaten by Harry Potter and the Cursed Child. But I was also up against 1984 in the category that I was in. But this was a play about the Battle of the Sexes tennis match that took place in 1973 in the Astrodome between Billie Jean King and Bobby Riggs. And it was about that, but it was also about the relationship between the sexes at the time and how far we've come and how far we haven't come in that sense. This is the theater that I produced this from. That's based here in Houston. We've renovated an old warehouse. We unfortunately opened it right before COVID and then had to close it again, but we're going strong again now. This is actually a work that I did, was getting prepared to do right before COVID, and this one was Pieces of the Moon, and this was a look at both the going to the moon and the anniversary that we were heading into for that, but at the same time going on in the United States was the Black Arts movement, and these two subjects are viewed very separately in people's mind, but they were actually happening at exactly the same time in the late 60s and early 70s, and there was an important conversation between those, and we addressed those issues within this play. We weren't actually able to produce this live. We actually ended up recording this in New York, mostly legally, during COVID, and then actually released it as a radio play for free during COVID. So and then lastly, this is a thing that we did last season that was really cool. This was called Plum Sugar. It's the story of Lauren Anderson, who was the first Black American to be a principal or prima donna ballerina for a major ballet company. This was in Houston in the early 80s, and it's the story of her life. One of the things that was really interesting about producing this play is that we got to work with the entire, almost the entire young and very talented cast of the Houston Ballet, and had tons of these people on our stage there. And I had no idea why these incredible artists all like to take headshots that make it look like they're naked, but apparently it's a thing. So with that, let's jump into the talk. So what we want to do today is talk about I think three big things. Number one is what happens to a tumor when it goes to pathology. Number two is how are sarcomas classified and graded, and number three, what are some of the genetic features of sarcomas? All right. So, so the first topic is what happens to my tumor tissue in pathology land, or the subtitle is A Fantastic Journey to Strange New Worlds. All right. So I just want to kind of walk you through what happens. So this is MD Anderson, where I work. We've actually, since I've taken these pictures, we've remodeled and it looks a little nicer. And I should probably retake all the pictures from this at some point so that you can see that we actually work in a slightly remodeled environment. But you'll get the gist of what happens. So on the other side of the window that's there in the top left, this is where all the surgical suites are at MD Anderson. Through this same area comes through if there's interventional radiology and we're just taking a sample for diagnosis, but everything kind of comes through that window and gets logged into the computer so that we know what patient it's associated with. The tumor tissue, then if it's a large resection, ends up getting examined by a pathologist. And we have a certain way that we look at it. We may correlate it with radiography, with the diagnosis, and then we cut it up so that we can actually see and take little pieces of it that help us determine all the features that we need to do to make both the diagnosis and to stage it. And we'll be talking about staging later. So ultimately, the tumor samples that we have are, we take little pieces of this and we put them in plastic cassettes for further testing. And it also, at this point, while the tumor is fresh, we're also taking it potentially if we want to do cytogenetics or for tumor bank, for research, or for molecular diagnosis. Electron microscopy we don't really use as much anymore. These tumor blocks are fixed in a chemical called formalin that sort of makes them more firm so that they're easy to deal with later, and you'll see why that's important. And they're loaded in a tissue processor overnight, and that's the thing here in the picture that looks like the little tiny washing machine. This has all kinds of different chemicals that go through it, but ultimately what we end up doing is sort of taking it to the point where we can, under pressure and heat, embed the tissue with wax, so that it's actually at this point really firm and preserved basically permanently, and that we can and that's easy for both long-term storage and for the additional steps that we need to do for diagnosis. So these are the little cassettes that come back out, and you can see here in the bottom. This is the now paraffinized or waxy little bits of tumor that are here. What we do next is actually take this little mold here and put more paraffin, and paraffin is essentially just more or less like candle wax. And so it melts and hardens actually at very reasonable temperatures. So we take hot wax, sort of put it in a mold, put the tissue piece in where we need it, and then freeze that back. And then on the back of the little cassette that was holding this, we're actually able to recycle that, and we actually put the little piece of tissue embedded in plastic on the top side of that. Right, so that's now the piece of tumor tissue there with the plastic that has all the patient identifiers that we need embedded in wax. Then we take it to something called a microtome, and with this, we're able to actually to cut little slices of this, almost like in some ways like slicing, like slicing a lunch meat super thin, with with one of the slicers at the deli. Except this is like super thin. You can see from the lower picture there that this is almost tissue paper thin, where it's almost translucent. You can almost see through the tissue. So this is really super thin, just a few microns actually in thickness. So that we can actually see the tissue really well, this is really clever. We actually float it on warm water, and when you float it on warm water, the paraffin really stretches out, and then you can pick it up on a glass slide. You can see here on the bottom, and then the tissue is spread out because of the heat. And then we can actually stain it, and we use pink and blue. So that we can actually, see what the tissue looks like, under the microscope. And there's some additional processing that's done in order to stain it with those. There's then a piece of glass that's put on the top, and then this is a slide here with the tumor tissue on it that I can then look at under the microscope in order to make the diagnosis. So all the paraffin blocks are saved. We can use them, in the future for either molecular diagnostics, most of which we can now do with paraffin tissue. We don't actually necessarily need frozen tissue for those kind of things, or if we need to cut additional slides to do other studies like immunohistochemistry to help us do the diagnosis, or if we want to use the the tissue for for research if the patient has consented for that. A lot of research is done by recutting this onto glass slides as well. So this is just some of the storage that we do now. I don't have this addressed here, but the cases now we actually, in addition to looking at them through the glass slides, we actually scan in and end up with electronic images of these as well, so that when we want to look at an old case from a patient, we actually have a scan of that so that we can look at it at the computer, so we don't have to pull the glass slides again. And that's actually really helpful. That's something called digital pathology. So let's move on now and talk about how sarcomas are classified and graded, or subtitle, do grades really matter in our postmodern world? So sarcoma classification can be really challenging. There's more than 50 histologic types, each have varying clinical, phenotypes and behaviors. There's a subset of tumors that are unclassifiable, but in the real world, benign entities are actually at least 100-fold more common than malignant, and some of these could actually be confused for sarcomas. So in addition to the 50 histologic types that are malignant, there's probably more than 100 that are non-malignant, and there can be some confusion here. And so because these tumors are very, very rare, it's uncommon for most pathologists to get a lot of experience with them. So there's only people at a few centers that actually see a whole lot of these, and it's not really that those of us that work at these centers are geniuses in terms of working with this. We just see a lot. We just see a lot of the tumors, and if you see things a lot, you become more of an expert than than someone who only gets to see something once or twice in a year. Some of the sarcomas that we work with, literally, some people will never make the diagnosis of that type of tumor ever in their professional lifetime, or maybe only see it once. But those of us that work at large centers will see all these things many to multiple times a year. All right. So now you can see it on the right. These are some pictures that are of the tissue that's gone all the way through the process that I described. And the sort of the first way that we look at this is, what does it look like under the microscope? And this gives us a lot of insight into the diagnosis. And we can initially look down the microscope and see, is it more round or epithelioid? Are they more elongated, which we call spindled? And this sort of allows us to start thinking about what type of category could this be in, and also gives us a sense from looking at it, does it have the sort of features that suggest that the case is malignant? Now, most sarcomas sort of fall into categories based on their histiogenesis or line of differentiation. So they sort of look like a normal connective tissue, or what we call soft tissue or bone in the body. And so we sort of name them after the tissue that they most look like. So osteosarcoma sort of looks like bone, and liposarcoma looks like fat. Various stages of fat. Angiosarcoma looks like blood vessels, but that doesn't work for all the sarcomas. There's some things like synovial sarcoma that that doesn't really look like any normal tissue. There is a tissue in your body called synovium, but synovial sarcoma looks nothing like that, and is actually not related to it. And that name just historically comes from the first cases being described near the knee, where there's a lot of synovium. Alveolar soft part sarcoma, again, there's not a normal tissue that comprises to. The same with Ewing sarcoma. And a lot of these that don't really match up well with normal tissue are the result of gene fusions that occur that cause the tumor to grow. And so they're driven by genetic pathways that normally don't occur in human beings. And I think this probably causes them to look very different than the other tissues that we have in our body. All right. So again, as we're looking at these under the microscope, we can see features that sort of may look like messed up versions of what the normal tissue looks like. So this is an atypical lipomatous tumor, or well-differentiated liposarcoma. It still sort of looks like fat, but it has some unusual nuclear features that allow us to make the diagnosis. This is a pleomorphic liposarcoma, another type of liposarcoma that we can pick up by seeing these really extensive lipoblasts in this background and make the diagnosis. Some tumors here, this is an alveolar rhabdomyosarcoma. Rhabdomyosarcoma means that it's like skeletal muscle, but honestly, this doesn't actually look a lot like skeletal muscle. And so we sometimes have to look at what type of protein expression that it has or genetic features of it in order to say, okay, that looks like that's showing differentiation like skeletal muscle and therefore is a skeletal muscle, tumor. So sometimes we have to use additional tools. So the classification of sarcomas is we use the WHO schema, and for the last 5 years, I've just recently, or 6 years, I've just recently rotated off, and we're finishing up the series now. I was on the WHO committee that was overseeing the classification of all of cancer. The third book that we did was soft tissue and bone tumors, and this is sort of the definitive classification that's used across the world for how we look at soft tissue and bone tumors. So these are the different iterations going back. This is the 5th edition, the 4th edition, and the 3rd edition. I worked on all 3 of these at different stages of my career, but this is the latest classification that we have. And so the book is sort of divided into 4 really big chapters. There's soft tissue tumors, and then there's the bone tumors. And then there's some things that these undifferentiated round cell sarcomas that can occur in either bone or soft tissue, and they kind of have their own separate chapter. And then we have a chapter on genetic syndromes that are associated with soft tissue and bone tumors. And that's sort of how we divide the classification. There are a lot of soft tissue tumors. This is the former list of all the ones that are currently recognized by the WHO. There's still new ones being discovered. And so when we're starting to plan the 6th edition of this book now, we already know that there's going to be some new tumors in there. But there's a lot. And then in addition to the soft tissue tumors, here's actually a whole page full of bone tumors as well. So there's really a lot of different types of these tumors. And this is what makes this field so complicated. But within the soft tissue and bone, within the soft tissue tumors, we have, primarily these 11 categories that everything sort of falls into. And these sort of really roughly correspond to the different types of soft tissue that you have in your body and the malignancies that can be associated with them. And then we have this final category, which are the tumors of uncertain differentiation, which is where we put all the tumors that show differentiation that don't clearly match up with a specific type of soft or connective tissue. All right. So that's kind of the classification scheme. And then how do we fit the, things and create all these categories? Well, we look at a lot of different factors for what determines whether something is a new specific type of tumor. We look at, clinical features and where they tend to arise, and the staging and the molecular, and what do they look like under the microscope, and what do we know about the pathogenesis, and all these different things. And all of these inform these specific diagnosis, and each different tumor type has characteristic features across all of these categories that allow us to differentiate it from the other tumors. All right. So the other thing that we're doing, in addition to diagnosing it, within a particular diagnosis, there are features that can tell us that the tumor may behave more aggressively or somewhat less aggressively for sarcomas. And this is what we call, in the soft tissue classification, the biological potential of the tumor. So now we're talking about soft tissue pathology in general. So that's including both malignant and benign forms. I'm going to kind of walk you through the categories that we have. So there are the benign tumors. These are not sarcomas. These are things like the most common one of these is probably a lipoma. They're not destructive. They don't metastasize. All right. And then on the far end, we have the fully malignant stuff, which are the sarcomas, like synoviosarcoma or liposarcoma or rhabdomyosarcoma that are fully malignant. But then we have 2 kind of weird categories in the middle that don't really fit into this dichotomy of either benign or malignant. And we call both of them intermediate, and they're either intermediate type 1 or type 2, or the type 1 means they're locally aggressive. So these are tumors that recur and they're infiltrative and they're destructive, but they don't metastasize. And a desmoid would be an example of that. So a desmoid is not benign because of how destructive it is, but it's not really malignant because it doesn't metastasize. And so this intermediate category is kind of the best place to keep it. The second type of intermediate category we have is rarely metastasizing. And so these are ones that are locally aggressive and they can metastasize, but they don't metastasize very often. They metastasize in less than 2% of cases. That's very different than most sarcomas, which metastasize in 10 to 20, even 50% or more of cases. And so there's a few things in there. Angiomatoid fibrous histiocytoma is one. Normally doesn't metastasize, but in less than 2% of cases, does. And so that's the category we use for that. But most things either fall into the benign or malignant, but there are categories in between. So the next thing, we have a grading system, and this works for most sarcomas, but not for all sarcomas. And this is for soft tissue, not bone. We have a way that we can look at, whether it looks like it's going to behave, more locally aggressively, or whether it's very likely to metastasize. And so this is grade 1, 2, or 3, or low, medium, and high. Grade 1 means it's much less likely to metastasize. Grade 3 means it's very likely to metastasize. And grade 2 means I don't really know whether it looks like it's going to be, more of a problem with local control or more of a metastatic problem. And so most of the time, grade 2 tumors get treated as though they were grade 3 tumors, because I can't tell you that it's unlikely to behave aggressively. All right. And the factors that go into this are tumor differentiation, which I'll talk to you about, mitotic count, which is how fast the tumor is growing, and then necrosis, which means is the tumor showing any evidence that it's actually dying. So here we have the differentiation score. This is for leiomyosarcoma, where if I look at it and it's hard to tell if it's even malignant, that's well differentiated. If I look down and go, that looks like smooth muscle and it looks malignant, that would be conventional and that would get a slightly higher score. And then there's some cases of leiomyosarcoma where I look at it and I have to, use immunohistochemistry or other tests to prove that it's a leiomyosarcoma, and it looks highly malignant, and that gets a higher differentiation score. Now, the other thing that we're looking for is how rapidly it's dividing. We do that by counting the number of mitoses. So this is literally, as a cell is dividing and the DNA is getting pulled apart, as it's replicated and going into two different cells, we can actually see that under the microscope. And that gives us a sense of how fast the tumor is growing or dividing. All right. And then lastly, this is necrosis, or the tumor dying, which is the final factor. And you think, oh, that must be great. My tumor's dying. That's a good thing, right? Well, it sounds like it's a good thing, but unfortunately what that means usually is that the tumor is growing so fast that it's outgrowing its local blood supply. And so it's so aggressive, it can't recruit enough blood vessels to continue to grow. And so while it sounds great that your tumor is dying, in this particular case, it means the tumor is growing so rapidly that it's very unlikely to be contained, and it's likely to spread and behave aggressively in most cases. So it's actually not a great sign. After you're treated, if you have a lot of necrosis, that can be a good sign that the treatment was effective. So by scoring across all those categories, we can come up with whether it's low, medium, or high grade, or grade 1, 2, or 3, and this actually correlates well for most tumor types, with survival. And this is just an example of this. This is metastasis-free survival, but overall, or sarcoma-specific survival shows a similar difference between the grades. And so you can see that if it's grade 1, this is this looks pretty good. This is if this line were to go straight out, that means everybody survives, and then down here is time, and this shows sort of over time how many people have an event that occurs. In this case, it's a metastasis. So you can see grade 1 is more favorable. Grade 3 is much less favorable, and the grade 2 is kind of in between. And grade 2, if we were better at dividing it, is a mixture of things that behave very well and things that behave very badly, but we don't yet have the tools to figure out which cases are which. All right. So this is another type of way that we look at how a tumor is likely to behave. It's a little bit different than grading. It's risk assessment, where we're using things other than things that we're looking down the microscope to use. And in this case, with gastrointestinal stromal tumor, we also look at where the tumor is. And because it's not using only anatomical variables, this is actually a risk assessment rather than grading, and is a little bit of a different system, but still kind of puts things into a low, medium, and high. It's a little more complicated than that, type of system. All right. So the next thing after grading is staging, and staging is just where is your tumor, in terms of where it started and has it spread anywhere else in your body, and basically how much is there. And so this is the eighth edition of the AJCC staging manual. This is something else that with a number of colleagues I worked on the committee to help create, and I just have little red stars by the pathologists that worked on this particular round. Usually it was just me and one other pathologist that was involved in this, but in this latest round we recruited a fair number of pathologists, actually a fair number of people across lots of different disciplines in order to be able to produce this latest edition. And we made a big change here. It used to just be one chapter on sarcoma, but we decided, sarcomas are so different from each other, it actually doesn't make sense to have just a single chapter and try to stage them all the same. And so we tried to come up with a system that broke it into different categories, which generally worked best with what anatomic site that they were arising in. And the staging systems are slightly different for each one of these, because the size of a tumor that may arise in the head and neck area has very different implications from the same size tumor that's maybe in the retroperitoneum. And this new structure, recognizes things like that. So this is for the extremity in the trunk, which is where we had the most data. We have, the tumors behave very differently from each other. And again, remember the upper lines on these types of curves are tumors that are behaving better. The lower lines are behaving worse. And so based on their size and their depth and different factors, there were different ways that they behaved. And by sort of combining these variables, we can have a prediction that allows us to predict how well a patient's likely to do based on the size of their tumor and whether it's spread or not. This is also important for when we're trying to analyze, outcome to treatments. We want to know that we're looking at patients that are likely to have behaved very similarly, so that when we compare patients who are treated and not treated, we can tell that the treatment is actually having effect, rather than maybe we have two groups where the patients actually are not comparable to each other. So staging is used in that sense as well. So now, just to quickly end, want to look at what are some of the genetic features of sarcomas, and this is really about cancer mutations and when your genes decide to go way off script, but not in a fun, improv comedy kind of way. All right. So here's sort of the broad genomic classification for sarcomas. We have those that have very simple karyotypes, which means when I look at the chromosomes initially by a karyotype, which is a very old-fashioned way of doing this, which we don't do anymore, they look fairly simple and normal. And then the other end of this are those that have really complex karyotypes. And then there's some stuff that's in the middle that's kind of intermediate. Some of the liposarcomas, particularly the well-differentiated and deep differentiated, are kind of between these two different models. But the simple karyotype ones very often are things that have translocations or gastrointestinal stromal tumor or desmoid, or where they just have a very small number of genomic changes that don't cause a lot of complexity with what the chromosomes look like. And then we have the complex karyotype sarcomas, which show tons and tons of changes in terms of what the chromosomes and DNA actually look like. All right, I'm going to show you some examples of this. So this is from the Cancer Genome Atlas that I co-led with multiple other people, where we looked at the genomic structure of a number of different types of sarcomas, and we were able to look at their mutations and the other alterations in DNA that cause the number of genes to change, how those genes are expressed into RNA, the methylation, which helps control RNA expression, microRNA, which is another way we control genetic expression, protein expression, then also clinical data. And by correlating all across these, we were actually able to find, some interesting features of sarcomas, which I'll talk about in a minute. This was part of the large TCGA project, which was the pan-cancer effort, where we compared, in the end, 33 types of cancer to see how they were alike and how they were very different. And I'll show you some data and some of the unique features of sarcoma within this. And this is another really big project that I and hundreds of other people worked on for more than a decade across all of cancer. This is the international consortium that we did with the Cancer Genome, Atlas, an international effort, and we combined all of our cases and sort of again looked across all of cancer. So this was the international effort. The TCGA was American led through the National Cancer Institute. So one of the things that we learned is by looking at the genetic expression of cancer, these are the 33 types that we looked at in the TCGA. You can see, and all the cancers are coded with unusual names, but I think you can see here in sort of the teal color, this is sarcoma, and that their genetic expression is really very unique from all the other different cancer types in terms of how they cluster. The one that was really close is another cancer that we actually wouldn't have thought was as closely related, but something called mesothelioma, that shows some relations to sarcoma and overlap. But everything else was, very distinctly different. And one of the big features that we noticed was that in terms of, alterations where we have increased numbers of copies of certain genes, that sarcomas were kind of the leaders in this. So this is all the TCGA across all the different cancer types, and all the things with these colored boxes at the right-hand side are the sarcomas. And these show the highest levels of copy number alterations, where we have, more repeated copies and losses of genomic features. And this is what makes the karyotype so complex. The exception to this was synovial sarcoma, which is an example of a simple karyotype sarcoma that really only requires one single translocation, or one change in the DNA in order to be fully malignant. And it was one of the most simple types of tumors. So that sort of shows you that, those categories there, and the differences between them. So just to conclude, pathology is absolutely necessary for a definitive diagnosis. Sarcomas come in many types, and they're often difficult or very challenging to diagnose because they're rare. And I would be happy to take any questions that anybody has, either about American and international theater, or sarcomas and sarcoma pathology. And by the way, it's a pleasure to be with you guys today. I just had my hip replaced 2 weeks ago, so I'm working from home. That's why I look a little rough. I will admit, giving this lecture in my pajamas, sitting in my home office. So, and I'm proud 2 weeks after having my hip replaced that I'm able to do anything at all. So thanks for inviting me.
Well, Dr. Lazar, thank you so much for taking the time today, especially after such a big surgery. Wow. It looks like we maybe have a couple questions. One is, what does the prefix mixo mean in when classifying a sarcoma?
Yeah, we just like to use a lot of fancy words like that to make it sound like what we're doing is really complex. So we have myxofibrosarcoma, and we have low-grade fibromyxosarcoma, and but no, what mixo means is that it has a lot of myxoid material. And myxoid material, not to be too disgusting, is kind of like a mucousy or boogery like consistency with the tumor that sort of happens, and it's a way that that proteoglycans or proteins are secreted and kind of pool in the tumor, and it has a fairly characteristic look under the microscope. It's called myxoid, but the consistency of it is kind of like mucus. So that's where that comes from. So if you have a myxoid tumor, basically you have a booger tumor. That's kind of a joke, but also kind of true.
Okay. I think one more question is, you talked about there are some types of tumors that are benign and malignant, and then there are some that kind of fit in the middle, like a desmoid, for instance. I know in other cancer types, you can have something that's benign and it can eventually turn malignant, like a polyp or something. Does that happen in sarcomas too? If it's benign, is it always benign?
Yeah, that's a good question. So most of the time that's true. Most of the things that are benign, like the lipoma, virtually never becomes a liposarcoma. And if that did happen, one should really go back and look at that initial diagnosis of lipoma, because it may have been a misdiagnosis initially, right? So that doesn't happen. There are a few cases where benign tumors can evolve to a malignant form, but those are very, very rare. And then there's some that are really interesting, and gastrointestinal stromal tumor is a good example of this, where if they're very small, they're very common. And in some people, they stay very small and they never grow large. And if they're below a certain size, they essentially are a benign tumor. But if they get bigger, then they can behave more aggressively, and sort of, where you draw that line and why some people's tumors grow to a certain size and stop, and, you never see them. Whereas other people's grow large and have a problem. We don't really know what the differences, are in that. There's a number of people that are trying to study that because it's an interesting problem. But those are some of the examples where there are things like that. But in most cases, things that are benign are generally benign, and and the things that are malignant. Now, unfortunately, the opposite doesn't happen either. We don't have a lot of benign tumors that become there are a lot of malignant tumors that become benign. So.
Right. Just one last question. So we have someone in here that was diagnosed with MIFS and was told it was low grade and slow growing, but is questioning, is there also a high grade version of this? So can you have low grade and high grade versions of all these various subtypes? I guess.
Yeah, with the myxo-inflammatory tumor that we were just asked at, generally that tends to stay lower grade. There are some cases that can become more aggressive, but that is very, very uncommon, and that one tends to be more of an issue with local control. But it doesn't really happen. It doesn't really matter what generally happens, right? What matters is what your case is like. And so, sort of following it and having a, careful examination, and, and following it clinically is very important to make sure that, what I can give you what the statistics are generally, but in your particular case, your tumor is either going to behave aggressively or it's not. So, the personal assessment there is important, but most of those tumors stay low grade.
Great. Well, thank you. I think we've
Yeah,
reached the
I
end.
see that there's a ton of
Okay.
other stuff in the chat, and I'm going to kind of turn my camera off, and I'm going to try to answer as many of these as I can
Oh,
from
thank you for that.
and if there's other stuff that people want answered, I'm pretty easy to find, and you can email me. Well, I'll just put my email in the chat, and if people have other stuff that they want to reach out and ask me. I'm a little slow answering my email because I'm technically on family medical leave right now, but I'll eventually get to your questions if you email them, and I'll throw my my email address in a second. And then I'm going to grade myself and grade that my lecture was outstanding, and I'm going to assume that everyone was very satisfied. And I recommend that everyone else do the same. So thank you very much.
Thank you so much, Dr. Lazar.